Hello, everyone. This is the second part of the important video on tips and tricks on the lymphatic, on the lymphatic and immune systems. And we're going to pursue our discussion about the thymus as a lymphatic organ. More on the thymus. As you know, we usually mention the lymph nodes as important organs in the lymphatic system that are concerned with filtration of the lymph and attacking the pathogens that are present in the lymph. But the thymus is not meant to filter the lymph, and it's not meant neither to trap the antigens, unlike the lymph nodes. The lymph nodes are bean-shaped structures that lie along the lymphatics. So when the lymph enters the lymph nodes that are full of lymphocytes, the pathogens get trapped there and can be attacked by the lymphocytes. But the thymus is designed to be a controlled environment for the development and the maturation of the T lymphocytes. We mentioned before in the previous part that the T lymphocytes, just like the B lymphocytes, are made in the bone marrow. And the bone marrow is the main site for synthesis and making of the blood vessels because it contains a hematopoietic cells or tissue that is concerned with a synthesis of the blood cellular components. But the T lymphocytes do not spend their whole life in the bone marrow. They leave the bone marrow via blood in order to reach the lymph. And why, in order to reach the thymus. And why do they want to reach the thymus? They want to reach the thymus because in the thymus, they will find the suitable environmental conditions for their maturation to be complete. And we talked about maturation in the last video. And we said that when the T lymphocytes are put in the suitable environment, they start developing their specific receptors, which we call the T cell receptors. At that time, they become mature, but they are still naive because they haven't yet encountered any antigen. So the immature T cells from the bone marrow enter the thymus via the blood, not via the lymph, like in the lymph node. And why isn't the thymus connected to lymphatics just like the lymph nodes, for example? This is because it's meant for the thymus to be isolated from lymph or from the afferent lymphatic vessels that carry lymph towards the thymus. So what connections then does the thymus have with the bone marrow? It is connected only via blood, but not via lymph. So the thymus is isolated on purpose. It is meant because it is meant for the T lymphocytes to stay in a rather closed environment, like in a bubble, to avoid exposure to external antigens until they are trained. This is exactly like when you are a medical student and you are not allowed to work in a clinic unless you get your certificate and then you get your license to work as a medical doctor. Before this, you just see clinical cases, you just attend rounds in the hospital, but you're not allowed to be fully responsible for treatment of a medical condition. So what would happen if the afferent lymphatics were in connection with the thymus and if they were allowed to bring in antigens randomly to the thymus? This would affect two things. When the lymphatics bring in antigens randomly, this will disrupt the positive and negative selection because, as you know, the positive and negative selection is determined according to the behavior of the T lymphocyte receptors, the T cell receptors, towards our self-antigens, which are the MHC1. And along all this time that is spent by the T lymphocytes in the thymus, the main idea is how the T cell receptor will bind with the MHC1. If it is bonded moderately, just to recognize and identify this MHC1 as self, and therefore to be tolerant towards it and to avoid killing it, then this cell is allowed to be released into the circulation, and it undergoes positive selection, as we said in the first part. But if the reaction or the bonding between the T cell receptor on the cell membrane of the T lymphocyte and the molecule that is MHC1 is too loose, then this cell will be discarded because it's not even capable of detecting self-antigens. And on the other hand, if the bonding is too tight, it's also discarded because this means that this is a dangerous T lymphocyte that is capable of attacking the even our cells and not only the pathogens. So it's not beneficial to the T lymphocyte to be exposed to antigens at that time, because at that time it is isolated from the rest of the body for one simple purpose, is to expose it to the self-antigens, to the MHC1 molecules that are present on the surface of our self-cell membranes, and to determine which one will be allowed to continue and will be labeled as positively selected or will be prevented and discarded and undergo destruction or apoptosis. The thymus does not produce lymphocytes. The thymus receives lymphocytes from the bone marrow, but however, it is considered as a primary lymphatic organ. Why? Because by definition, a primary lymphatic organ is an organ where the lymphocytes originate and or mature into immunocompetent cells. So in the case of the thymus, lymphocytes do not originate in the thymus, but we have the other option, which is, or they mature into immunocompetent cells. What are immunocompetent cells? They are cells that are capable to recognize the specific antigen and to attack it and get rid of it. The thymus thus qualifies as a primary lymphatic organ because when the T cell precursors reach the thymus from the bone marrow, they are still immature. And inside the thymus, they mature after having spent a certain period of time inside the thymus. And during that period of time, there will be positive selection for cells that can identify or recognize the MHC1 molecules as self, or they are negatively selected and discarded and undergo apoptosis if they are overreactive against self cells, because in such a case, if they are released in the circulation, they could result in autoimmunity. Why isn't the thymus capable of producing lymphocytes from scratch? Because it does not produce as many or as abundantly the stem cells like in the bone marrow, and therefore it's not capable of creating those T cell precursors from nowhere. But as you know, the environment is very important for the development of traits or characteristics. So even if the T lymphocyte has the genes for formation of the T cell receptor, if it stays inside the bone marrow, it will not find the suitable environmental conditions that allow those genes to be translated and that would allow the receptors, the TCRs, or T cell receptors to form on the cell membrane of the T lymphocytes. But it's only the thymus that is capable of providing this favorable environment for those genes to be expressed, for the TCR molecule to be formed on the surface of the T lymphocyte, and for the immature, naive T cell precursors to develop into mature and immunocompetent cells. So to conclude, the bone marrow is capable of producing not only T lymphocytes, but B lymphocytes, but they are at the very beginning of their lives. They are called B and T cell precursors. And the thymus, on the other hand, this is the thymus, we should have written here, the thymus. The thymus helps the maturation of the T cells after receiving the necessary education. So the thymus acts like a school for the T cells to get mature, educated, and immunocompetent. And that's why, although it does not apply or it does not fit into the first condition, which is production of lymphocytes, yet it fits into the other one, which is teaching or educating the T lymphocyte. So the bone marrow is like a factory that produces the T cell precursors, and the thymus is like a school that receives those T cell precursors and teaches them how to become immunocompetent. On the other hand, the lymph nodes and the spleen is a very large lymphatic organ that lies behind the stomach, and it is connected to afferent lymphatics that carry lymph towards the spleen, and efferent lymphatics that carry lymph away from the spleen. On the other hand, the lymph nodes are present in groups all along, like for example, the group in the neck, the cervical lymph nodes that are present all along the pathway of the lymphatics. The lymph nodes and the spleen are considered secondary lymphatic organs. They do not make lymphocytes from scratch, and meanwhile, they do not teach them how to become immunocompetent. They just receive them after having been mature and immunocompetent. But the importance of the secondary lymphatic organs is that they are the battlefields of the lymphatic system. They receive the foreign antigens and they have the necessary immunocompetent T and B lymphocytes that can attack those antigens. So in a primary lymphatic organ, the lymphocytes are produced if it is the bone marrow, or are not produced if it is the thymus. But what's common is that they mature inside the primary lymphatic organ. As you know, the T cells mature in the thymus, whereas the B cells mature in the bone marrow. And they do not only mature or get mature, but they also become immunocompetent. What do we mean by immunocompetent? There are 3 steps for a cell to be immunocompetent. The first one is, or 3 conditions. The first one is that it has to recognize its specific antigen. And in order for it to recognize the specific antigen, it should have acquired its receptor, the T cell receptor, and it should also be ready to respond to the antigen. Of course, acquiring the T cell receptor and being able to recognize the specific antigen, I mean the first 2 steps are achieved in the thymus. But in order for them to be ready to respond to antigen, they should be activated. So an immunocompetent cell is still naive because it hasn't yet encountered the specific antigen. Furthermore, it still needs to be checked by the immune system using the MHC1 molecules. Will it be immunotolerant towards them and therefore allowed to work more and be activated and be released into the circulation, or would it be overreactive or non-reactive overreactive or cannot bind at all with the MHC1 molecule, in which case it would be discarded and undergo death and apoptosis. In the secondary lymphatic organs, this is like a recapitulation of what we said before. The lymphocytes, it is the site where the lymphocytes encounter the specific antigen. It is the site where the lymphocytes become activated by certain chemical substances, and it is the site where the lymphocytes, after having received those signals, will undergo proliferation or cell division. And when they proliferate, as you see, they give a clone of cell, a group of cells that are all similar, that all have the same surface receptors or the same surface molecules. And because they are exact copies of each other, they form what we call a clone, and this is what we call clonal proliferation. After having proliferated, they will divide themselves or categorize themselves. They will differentiate. So they will divide themselves into cells that act on the spot immediately. So the effector cells act immediately upon finding an antigen, and these are the effector cells. And the memory cells, on the other hand, are kept for the future. They stay in the body, they remain circulating in the body. And as we said in part 1, they are responsible for attacking the antigen upon any secondary exposure. And their response is called the secondary anamnestic response, anamnestic response. This means that the memory cells, there is no amnesia. They do not forget the antigen. They remember it, they act very effectively and in a very short period of time. The effector cells act on the spot, and they are categorized into two groups depending on the surface molecule on top of the cell membrane. If the surface molecule is a CD4 plus molecule, then they are helper cells. They help other T cells and other B cells to be activated, and they awaken the cells, and they warn the body that there is like an antigen that is threatening the body. But if they have the CD8 plus molecule, then they are cytotoxic cells, and upon encountering any labeled stressed cell, for example, they will release cytokines or chemical substances to kill this cell. In this very short table, we are comparing and contrasting maturation and activation. According to what we have said before, maturation should occur in a primary organ, whereas activation, proliferation, and differentiation will occur in a secondary organ. Maturation is more related to the development of the surface receptors on the surface of the molecule, whether they are T cell receptors on top of the T lymphocytes that develop or that are acquired in the thymus, or B cell receptors on top of the B cells acquired in the bone marrow. But activation, on the other hand, necessitates antigen exposure and at the same time receiving activation signals from other cells like the helper cells. In case of maturation, the development of these receptors and their interaction with the MHC1 molecules establishes self-tolerance and allows the body to differentiate between cells that are not reactive against their own MHC1 molecules, and therefore that are positively selected and allowed to be released into the circulation, or cells that are not self-tolerant and that should be discarded. So we are developing immunocompetent cells, but naive cells. Why naive? Because we haven't yet encountered the antigen, and therefore we haven't yet entered into the phase of activation. But in activation, the cell is no more naive. It has been exposed to the antigen. It has proliferated and has shown clonal expansion and has also differentiated into effector and memory cells. The last thing to say is that, to summarize, in a primary lymphatic organ, there is no enemy present, and therefore the function of the primary lymphatic organ, besides formation of the cells, is to provide a safe environment for the cells to be taught. While in a secondary lymphatic organ, like in the lymph nodes and the spleen, the enemy is there. There is exposure to the antigen, and this is when alertness and efficiency is most needed. The last table to discuss is between primary and secondary lymphatic organs. Is the antigen present in the primary? No, there is no exposure to the antigen in the secondary organ. Yes. Does maturation occur there? In the primary lymphatic organ, yes, it is the place and the time when maturation occurs. In the secondary, no, there is no time to wait for the cell to become mature. It is received mature. Is there activation in the primary lymphatic organ? Of course not, because there is no exposure to the antigen, and activation requires antigen exposure, so it occurs in the secondary lymphatic organ. Of course, because there is activation, then proliferation here will be massive, whereas proliferation here is very slow. The main function of the primary lymphatic organ is education, whereas the secondary lymphatic organ's main function is to elicit an immune response. By having said this, we would have finished the tips and tricks on the thymus, part 2, and see you in another video.